Good morning, students. Today we are going to learn that how to introduce an alien piece of DNA or any foreign DNA or recombinant DNA into host cells. So this is the topic that what we are going to learn today, that how to introduce an alien piece of DNA or recombinant DNA or plasmid DNA into the host cells. So there are the different host organisms. We can use the bacterial cells also as the hosts. Animal cells can also be used as host. Plant cells can also be used. So how to introduce the alien piece of DNA or recombinant DNA into the host cells? Now, first question is why to introduce into the host cell? So actually, when we introduce a recombinant DNA or any alien piece of DNA into the host cells, this is the transformation of the host cells, and then we use that host cells at industrial scale production of the any product. So first of all, we will talk about the how to introduce a plasmid DNA or a bacteria or a recombinant DNA into the bacterial cells. So suppose we have the medium, culture medium, because bacterial cells are very small and their growth rate is very high. They multiply in every 20 to 30 minutes they multiply. So that's why to introduce the alien piece of DNA or recombinant DNA or plasmid DNA, we use the culture medium of bacteria, and in the culture medium there are the many bacterial cells. So we use the many recombinant DNA also. So suppose we add this recombinant DNA or plasmid DNA in this culture medium of the bacteria. But the problem is that this DNA is the negatively charged molecule first of all. Second thing is that it is the hydrophilic molecule. DNA molecules are negatively charged first of all. Second thing is that it is hydrophilic molecule, while you know that the plasma membrane of the cells, plasma membrane consists of lipoprotein, lipid and protein. There is the bilayer of lipids, phospholipids, and that bilayer of lipid or phospholipid makes the hydrophobic layer. So a hydrophilic molecule cannot easily cross the hydrophobic layer. Likewise, because this DNA molecule is the negatively charged, means the polar. There is the charge, negatively charged means this is the polar, while the plasma membrane there is the lipid bilayer and that is the non-polar. So a polar molecule cannot cross the non-polar layer. So then how this recombinant DNA or plasmid DNA will enter into these bacterial cells? So what we do, we have to make these bacterial cells competent. We have to make these bacterial cells competent to take up the DNA from the medium. How to make competent? What we do? So actually there are the two processes to make these bacterial cells competent to take up the DNA from the medium or plasmid DNA from the medium. First of all, we do the treatment by calcium ion, or we can use any cation. We can use any cation or calcium ion. We do the treatment of these bacterial cells by any calcium ion or any cation we can use. And this cation or calcium ion now increases the efficiency. It increases the efficiency with which DNA enters. It increases the efficiency with which DNA enters into the bacterial cells through the pores present in the cell wall of the bacteria. So what is the first process what we do to make these bacterial cells competent to take up the DNA from the medium? So first of all we do the treatment of these bacterial cells with any cation or calcium ion, which increases the efficiency with which DNA enters the bacterial cells through the pores present in the wall of the bacteria. Actually, what we do, actually this calcium ion, this is the positively charged, while the DNA is the negatively charged. So this calcium ion attract the negatively charged molecules towards the bacterial cell, first of all. So this calcium ion, this is the positively charged, or any cation we can use that attract the negatively charged molecule of DNA towards the bacterial cells and increases the efficiency with which the DNA enter through the pores of the plant cells or bacterial cells. Cell wall, the pores present in the cell wall. Now the second step, what we do? So first of all, the treatment with the any cation or calcium ion, and the second one is the heat and shock treatment. What we do in the second step? Now after the treatment of these bacterial cells with the calcium ion or any cation, after that now we add this recombinant DNA in the medium, and then what we do? We now place this culture medium place on the ice. This is the cold treatment. First of all, what we do, that we place or we incubate these bacterial cells along with this recombinant DNA on ice for some time. After that, again we transfer this medium, these bacterial cells along with this recombinant DNA or plasmid DNA on heat 42 at 42 degrees centigrade. This is the heat treatment. So first cold treatment, then again heat treatment, and you know that the heat expanses and cold contract. So this is the contraction and expansion of these bacterial cells. Again we transfer these bacterial cells with this recombinant DNA on ice, means again the cold treatment. So in this way, alternately we do the heat and cold treatment that helps the bacterial cells to take up the DNA from the medium. So this is the process to make the bacterial cells competent to take up the DNA from the medium or recombinant DNA from the medium. So this is the one thing now we talked about these bacterial cells, that how to introduce the alien DNA into the bacterial cells. We learned that the first of all we do the treatment with the calcium or any cation that increases the efficiency. Second, we learned that the heat and cold treatment, alternately heat and cold treatment, that also helps the bacteria to take up the DNA from the medium. Now we talk about the animal cells, that how to introduce the alien piece of DNA into animal cells. So animal cells, you know that the size is large sized animal cells, and the growth and division process is also slow than the prokaryotic cell, because these are the eukaryotic cell. Here first of all we learned about how to introduce the DNA into the prokaryotic cells. Now we are going to learn in the eukaryotic cells. The animal cells are the eukaryotic cells. So in the animal cells we can introduce the alien piece of DNA or plasmid DNA or recombinant DNA directly. We can directly inject the recombinant DNA into the nucleus of animal cell through the micro injection or micro pipette. Here the micro pipette used in the micro injection, and through this micro pipette or micro injection we can directly inject the recombinant DNA or alien piece of DNA into the nucleus of animal cells. So for animal cells we learned. Now we talk about the plant cells, that how to introduce the recombinant DNA into the plant cells. So what we do to introduce the recombinant DNA or plasmid DNA into the plant cells? What we do? Plant cells are bombarded with a high velocity, with the gold and microparticles of gold and tungsten. What we do, we bombarded the cells, plant cells, with high velocity microparticles. We bombarded with high velocity microparticles of gold and tungsten coated with this recombinant DNA. Coated with this recombinant DNA. This helps to introduce the recombinant DNA into the plant cell, and this is called the biolistic or gene gun. So this is the gene gun, means through the gene gun we can introduce the alien piece of DNA into the plant cells. So what we learn in the plant cell, what we do, the plant cells are bombarded with high velocity, the microparticles of gold and tungsten coated with the recombinant DNA. That helps to introduce the DNA into the plant cell, and the process is called the biolistics or gene gun. Now we talk the another also a method we can use to introduce the alien piece of DNA or recombinant DNA into the plant cell, animal cell or bacterial cells. So the fourth method that we can use is the disarmed pathogen vectors. Disarmed pathogen vectors can be used to introduce the plasmid or alien piece of DNA into the plant cell or animal cell. For example, we can use Agrobacterium tumefaciens. We have already studied the Agrobacterium tumefaciens. We know that the Agrobacterium tumefaciens is the natural genetic engineer that introduced the tumor-inducing DNA into the host cell. And through that tumor-inducing DNA we can introduce the our desired gene also. So the disarmed means to make it inactive or non-pathogenic. So disarmed means to make the bacteria or viruses non-pathogenic. And through this disarmed pathogen vectors we can introduce the alien piece of DNA or plasmid DNA into the plant cell or animal cell. For example, what we learned that through the Agrobacterium tumefaciens we can introduce our desired gene into the plant cells. Likewise, there are also many retroviruses. We can disarm the retroviruses after making non-pathogenic. Through that retroviruses we can introduce the desired gene into the animal cells. So for the animal cells we can use the retroviruses. For the plant cells we can use the Agrobacterium tumefaciens, means the bacterial cells, Agrobacterium tumefaciens. Likewise, through the bacteriophages also we can introduce in the bacterial cells, bacteriophages also. So these are the different methods that how to introduce recombinant DNA or an alien piece of DNA or plasmid DNA into the host cell for the transformation of the host cell, to use that host cell in the industry for the large scale production of our desired product. Thank you.